Hello everyone, welcome to my channel Anupma Biology Classes. In this video, our topic is photosynthesis in higher plants. This video is taken from Plant Physiology series, CBSE class 11th, part 1st of chapter 14. Now, let's see here the lecture overview. So in this video, we will see definition of photosynthesis, equation of photosynthesis, magnitude of photosynthesis, chloroplast, the site for photosynthesis, the chloroplast pigments in which chlorophylls, carotenoids and phycobilins. Now let's start the topic, and here the first thing you will have to know in detail that what is photosynthesis. So, photosynthesis is a physiochemical process by which photosynthetic organisms convert light energy into chemical energy in the form of reducing powers as NADPH and ATP, and use these chemicals to drive carbon dioxide fixation. So, the photosynthesis, which is taken from a Greek word that is photon, which means light, and the synthesis, which means putting together. So photosynthesis is the most important anabolic process on earth by which green plants, which means autotrophic organisms, synthesize complex carbohydrates from simple substances like the carbon dioxide and water with the help of light energy and purify the atmospheric air by consuming carbon dioxide and evolving oxygen. In other words, carbon dioxide of atmosphere is fixed into carbohydrates and the light energy is converted into the chemical energy. So, the process of photosynthesis is the transformation of photonic energy into the chemical energy by the green parts of the plants. Now, the question is, what are photosynthetic organisms? So now see here for this, on the basis of generation of the oxygen during photosynthesis, the photosynthetic organism may be oxygenic or anoxygenic. So photosynthetic organisms are of two types, oxygenic and anoxygenic. Oxygenic photosynthetic organisms include both eukaryotes as well as prokaryotes. So if we talk about the oxygenic prokaryotes, so these are the cyanobacteria, and if we talk about the eukaryotes, these are plants and the photosynthetic protists. And in case of anoxygenic, here you can see only prokaryotes, which are the green and purple photosynthetic bacteria. So see here, these are the prokaryotes, which is oxygenic photosynthetic prokaryotes, which is cyanobacteria. Now these are the plants and photosynthetic protists. So these are the eukaryotic oxygenic photosynthetic organism. And see here, that is the purple photosynthetic bacteria and the green photosynthetic bacteria, which are the prokaryotes and anoxygenic photosynthetic organism. Now let's see here, that means photosynthesis is a two-stage process. So in the first stage here, the process which is known as the light dependent one, that means light reaction, that requires the direct energy of the light to make energy carrier molecules that are used in the second process. And this second process is Calvin cycle, which is known as light independent process, occurs when the product of the light reaction are used in the formation of carbohydrate. So in photosynthesis there are two processes. First one is the light reaction and the second is dark reaction, which is known as Calvin cycle. So in case of light dependent reaction, here you can see that in presence of light, and in case of Calvin cycle, which is known as light independent reaction, here lights doesn't require. Now after this, what are the equation of the photosynthesis? The simple overall equation usually given to represent photosynthesis is as follows. That means 6CO2 plus 6H2O in presence of light and chlorophyll forms C6H12O6 plus 6O2. The This equations represents early model of the photosynthesis when it was presumed that the molecular oxygen came from carbon dioxide and not from water. It was von Neil in 1931, a graduate student, who observed that during bacterial photosynthesis, hydrogen sulfide was broken down to hydrogen and sulfur. The hydrogen reduces the carbon dioxide to synthesize carbohydrate, and sulfur is accumulated as a waste product. Now in this equation here you can see that means 12H2S plus 6CO2 in presence of light and the bacterial chlorophyll form C6H12O6 plus 12S plus 6H2O. This led to the postulation that in the higher plants, water is utilized in the place of H2S and the oxygen is evolved in the place of sulfur. Nearly 10 years later, this hypothesis was confirmed by the isotopic studies made by Rubin and Kamen in 1941. They used the radioactive isotope of oxygen in water and found that the oxygen liberated in the process was isotopically leveled. On the contrary, when oxygen 18 in carbon dioxide was used, the evolved oxygen did not show radioactivity. Thus, the overall equation of the photosynthesis is currently accepted as here, that means 12H2O plus 6O2 CO2 in presence of light and chlorophyll forms C6H12O6 plus 6O2 plus 6H2O. So photosynthesis, here you can see the complete equation. Now after this, what are the magnitude of the photosynthesis? So if you talk about the magnitude, concentration of gaseous carbon dioxide in the atmosphere is about 0.03% by volume. At this rate, the atmosphere of our Earth contains about 2200 billion tons of carbon dioxide, which is sufficient to support the photosynthesis for a few hundred years, even if no further amount is added. The carbon dioxide present in the oceans in the form of dissolved gas or the carbonates is estimated about 11.2 into 10 to the power 14 tons, which is more than 50 times of the amount present in the atmosphere. Green plants fix about 70 to 80 billion tons of carbon dioxide annually by the process of photosynthesis. Out of total carbon dioxide fixed, about 90% is fixed by the water plants, mainly in oceans, and less than 10% is fixed by the terrestrial plants. Curiously, only 0.2% of the total solar energy falling on Earth's surface is utilized by the photosynthetic organism. Yet, this amount of trapped energy is just sufficient to meet the entire food requirement of all living organisms on Earth. Now, after this, chloroplast, which is the site for the photosynthesis. So, what are the chloroplast? Chloroplast, which is taken from the Greek word that means chloros, which means green, and plastos means molded, are the green plastids which occurs in all the green parts of the plants. They are actual site of the photosynthesis. Here you can see that the structure of the chloroplast, the maximum number of chloroplasts occur in leaves with over 500,000 per square millimeter. They occur mostly in the chlorenchymatous cells of leaves and young stems. Each mesophyll cell may contain as many as 300 chloroplasts in its peripheral cytoplasm. In algae and bryophytes, they occur in almost all the green cells. Now, algal chloroplast vary in shape and size. Here you can see the different shapes of the chloroplast. There is found in case of Spirogyra, collar shaped in Eulothrix, star shaped in Zycnema, cup shaped in Chlamydomonas, bell shaped in Chlorella, and reticulate in Oidogonia. So the chloroplast of higher plants are usually discoid, ellipsoidal or biconvex lens shaped. They are usually 4 to 10 micrometer in diameter and 1 to 3 micrometer in thickness. Each chloroplast of the hair plant is an organelle with an outer envelope consisting of a double membrane which acts as a selective barrier to the movement of cellular metabolites into or out of the chloroplast. The chloroplast envelope encloses a liquid proteinaceous matrix called stroma. Here in this picture you can see the structure of the stroma, that means aqueous fluid. The whole liquidy medium of the stroma is hyaline, slightly electron dense, granular matrix and contains all the necessary enzymes of the photosynthesis. It also contains 70S ribosomes, circular DNA and RNA. The chloroplastic DNA is called plastidome. The stroma is the site of dark reaction of the photosynthesis. The lamellar system within the stroma forms flattened sac-like lamella called thylakoids. Here you can see that the structure of the lumen inside the thylakoid regions, that means the thylakoid completes a stacked structure. These thylakoids are stacked in some places to form grana. The grana stacks are interconnected by the membranous lamella called stroma lamella or Fred channels. The space within each thylakoid is connected to the space within every other thylakoid, forming a common thylakoid space. The major function of the thylakoids is the perform photosynthetic light reaction. The pigments and other factors of the light reactions are usually located in the thylakoid membranes. Cyanobacteria and other photosynthetic bacteria do not possess chloroplast. However, the photosynthetic pigments of these organisms are located on thylakoids which lie freely in the cytoplasm, and their photosynthetic pigments are also different from those of eukaryotes. If we talk about the location of photosynthetic pigments in the chloroplast, the major photosynthetic pigments are the chlorophyll A, chlorophyll B and carotenoids. It has been shown that the chlorophyll molecules are non-covalently associated with the protein forming pigment protein complexes. They are located in the thylakoid membranes of the chloroplast. Now, there are four types of complexes. Here you can see that photosystem first, that complex, photosystem second complex, cytochrome B6F complex and ATPase complex. The pigment molecules associated with photosystem first complex are located in the non-appraised part as well as the stroma thylakoids, whereas those associated with the photosystem second complex are located more towards the appraised regions of the grana thylakoids. So here you can see that diagram which showing the lateral distribution of photosystem first, photosystem second and ATPase complexes between appraised and non-appraised thylakoid membranes. Now, the chloroplast pigments. So, what are the chloroplast pigments and how many? Pigments are the organic molecules that absorb light of a specific wavelength in the visible region due to the presence of conjugated double bonds in their structures. The chloroplast pigments are fat soluble and are located in the lipid part of the thylakoid membrane. There is a wide range of chloroplastic pigments which constitute more than 5% of the total dry weight of the chloroplast. They are grouped under two main categories, which are the chlorophylls and carotenoids. The other photosynthetic pigments present in some algae and cyanobacteria are phycobilins. So the first one, that is the chlorophyll. Chlorophyll in Greek, chlor means green and the phyll means leaf. So chlorophylls are the green photosynthetic pigments present in all photosynthetic autotrophic organisms. There are about 10 types of chlorophylls: chlorophyll A, B, C, D and E, bacteriochlorophyll A, B, C and D, and bacterioviridae. Of all, only two types, that is chlorophyll A and chlorophyll B, are widely distributed in the green algae and the higher plants. Chlorophyll A is found, here you can see the structure of the chlorophyll A, found in all the oxygen evolving photosynthetic plants. It occurs in several structurally distinct forms which perform distinct roles in the photosynthesis. It directly takes part in the photochemical reaction, hence it is termed as primary photosynthetic pigment. Other photosynthetic pigments, including chlorophyll B, C, D, and E, carotenoids, and phycobilins, are called accessory pigments. Because they do not directly take part in the photochemical act, they absorb a specific wavelengths of the light and transfer energy finally to the chlorophyll A through electron spin resonance. So the structure of the chlorophyll molecule was first studied by Willstetter, Stoll and the Fischer in 1912. Each chlorophyll molecule shows a tadpole-like configuration consisting of a porphyrin head and a phytol tail. The porphyrin head is made up of porphyrin system in which four pyrrole rings linked together by the methane group form a ring system. The center of the tetrapyrrole, which is also known as pyrrole, is occupied by a divalent magnesium which is complexed with the nitrogen atoms of the four pyrrole rings. Each chlorophyll has a fifth ring containing only carbon atoms. The porphyrin ring has several side groups which specify the properties of the pigment molecules. The phytol tail is made up of 20 carbon alcohol and bound in ester linkage to the fourth pyrrole ring. Chlorophyll C here, the phytol chain is responsible for the lipoidal solubility of the chlorophyll. So the chlorophyll B differs from the chlorophyll A in having an aldehyde, that is CHO group, instead of methyl, that means CH3 group, on the ring second of the porphyrin head. Now, what are the main difference between the chlorophyll A and chlorophyll B? The empirical formula of chlorophyll A is C55H72O5N4Mg, while the chlorophyll B empirical formula is C55H70O6N4Mg. Chlorophyll A is blue-green in color, while the chlorophyll B is yellow-green in color. Chlorophyll A is the primary pigment, while the chlorophyll B is the accessory pigment. Chlorophyll A has a methyl group attached to the third carbon, while the chlorophyll B has an aldehyde group attached to third carbon. Chlorophyll A is soluble in the petroleum ether, while the chlorophyll B is soluble in methyl alcohol. So these are the main difference between the chlorophyll A and B. Now, classification of algae based on the types of pigments. Here you can see there's some taxonomic groups and the photosynthetic pigments. So in case of the cyanobacteria, chlorophyll A, chlorophyll C, phycocyanin and the phycoerythrin present. While in case of chloroxybacteria, chlorophyll A and chlorophyll B present. In case of green algae, that means chlorophyta, chlorophyll A, chlorophyll B and carotenoids present. While in case of red algae, which means known as rhodophyta, chlorophyll A, phycocyanin, phycoerythrin and phycobilins present. In case of brown algae, that means phaeophyta, chlorophyll A, chlorophyll C, fucoxanthin and other carotenoids present. While in case of golden brown algae, that means chrysophyta, chlorophyll A, chlorophyll C, fucoxanthin and other carotenoids present. In case of dinoflagellates, that means pyrophyta, chlorophyll A, chlorophyll C, peridenine and other carotenoids present. And in case of vascular plants, chlorophyll A, chlorophyll B and carotenoids present. So here you can see the classification of the photosynthetic pigments in the taxonomic groups, that which type of photosynthetic pigments are present in which taxonomic group. Now, after this, the accessory pigments. So the first one, which is the necessary, are the chlorophylls. After this, the accessory pigments are, the first one is the carotenoids. So what are carotenoids? Carotenoids are the yellow, brown, orange or reddish pigment usually found in the close association of the chlorophyll in all photosynthesizing cells. They occur in thylakoids and act as accessory pigments of the photosynthesis. They absorb light energy in the mid region of the visible spectrum and transfer their absorbed energy to the chlorophyll molecules. They pick up nascent oxygen released during the photooxidation of water and change them into the molecular state. So, thus they protect chlorophyll molecules from the photooxidation. The carotenoids also occur inside the chromoplast in flowers and fruits and make them conspicuous to animals for the pollination and dispersal. So here you can see the 5 essential carotenoids, which are the lutein, alpha carotene, beta carotene, lycopene and zeaxanthin. So in case of carotenes are usually, in general, there are two major groups of carotenoids, the carotenes and the xanthophylls, and both of them are soluble in the organic solvents. So carotenes are usually hydrocarbons with a general formula of C40H56. They absorb blue and green lights and appear yellow and red in color. The most common carotene is beta carotene, which is converted to vitamin A by the animal and human beings. Next is xanthophyll. Xanthophylls are oxygen derivatives of the carotenes containing 1,8 oxygen atoms. Lutein is responsible for the yellow color in the autumn foliage. Now, after this, see the phycobilins. The phycobilins constitute a major group of photosynthetic pigments occurring in blue-green algae, cryptomonads, and the red algae. They are protein-linked pigments destroyed by heat. These water-soluble pigments are thought to be localized in small granules called phycobilisomes attached to the thylakoids. They are useful in chromatic adaptations like chlorophyll. These pigments are open tetrapyrrole but do not contain magnesium and phytol chain. So they absorb light and help in photosynthesis as accessory pigments. There are two types of phycobilins. First one is the phycocyanine and the second is phycoerythrin. So blue-green algae have more quantity of the phycocyanine, whereas red algae have the more phycoerythrin. After this, now the photosynthetic organism and the location of the photosynthetic pigments. So the first one, that is the oxygenic photosynthetic organism. In case of eukaryotes, thylakoid membranes of chloroplast. In case of prokaryotes, thylakoid membranes. In case of anoxygenic photosynthetic organism, in green photosynthetic bacteria, chlorosome and plasma membrane, and in case of purple photosynthetic bacteria, chromatophores of plasma membrane. So this is the end of video in which we knew about the photosynthesis, its equation, its magnitude, chloroplast, chlorophyll, carotenoids and phycobilins. The next upcoming video will be on the important terms of the photosynthesis. So, if you understand this video, please like and share it and subscribe to my channel Anupma Biology Classes. If you have any questions, any queries or any suggestions, you can ask in the comment section below. Thank you for the watching.